First of all, I would like to thank the Creutzfeldt-Jakob Disease Foundation for funding this research. The title is Exploring the Zoonotic Potential of Porcine-Derived Materials from Animals Exposed to Infectious Prions. And this specific research is part of a larger program we are running in my laboratory, trying to assess the potential role of different animal prion diseases. So this is the disclosure for this presentation. And as you may know, prion diseases affect humans and animals. In humans, we can consider prion diseases as rare diseases. The incidence is low, affecting, in average, 1 person per million per year. And the most common disease in humans is Creutzfeldt-Jakob disease in its sporadic form. But in animals, prion diseases can reach epidemic proportions. For example, the bovine spongiform encephalopathy or mad cow disease was a big problem, especially in Europe at the end of the last century. Scrapie affecting sheep and goats also can have a very high prevalence. And an important problem, at least in the United States, is chronic wasting disease that affect captive and wild cervids from different species. So one of the main concerns regarding prion diseases involve their zoonotic potentials. For example, it's well established that BSE, or mad cow disease, can generate what we call variant Creutzfeldt-Jakob disease in humans. And it is hypothesized that BSE was originally generated by sheep scrapie, although this is not completely proven. But on the other hand, sheep scrapie is also considered to be not so much of a threat for human disease. Some evidence suggests that some specific strains of scrapie could affect humans, but this is still under investigation. So there are some, well, not some, but several paradigms involving the zoonotic potential in the interspecies transmission of prions. Like, for example, what happened, for example, if the BSE material comes back to sheep. This has been experimentally done, and it's known that the strain, the agent that is generated, is different from scrapie. And the properties of this material has unprecedented features, and this is some of the things that we are investigating in the laboratory. Well, also, as you may know, the central event in prion diseases involved the misfolding of a normally folded protein that all of us produce in our bodies that we call PRPC, prion protein cellular in its cellular form, that under certain circumstances change its conformation, it misfold, in what we call PRPSC from prion protein scrapie. So the process of protein misfolding involving the prion protein follows this kinetic. So the limiting step for the misfolding is the generation and the stabilization of a small aggregates that have this abnormal conformation. Once these particles are stabilized, they will start to recruit normally folded protein into the growing aggregates, and these particles actually will get together and they will deposit in the brain of affected individuals, generating eventually prion diseases. So we know that the limiting step, again, for the generation of misfolded prions, the accumulation of misfolded prions in tissues, it can be substantially decreased if we add preformed aggregates or seeds into the system. And actually, this explains very well the process of infectivity that we observe in prion diseases. So in order to investigate this central event in prion diseases, the conversion of the prion protein and subsequent aggregation, we have several tools. One of the best is the use of animal models, specifically transgenic animal models. Basically, in here, what is done is to remove the endogenous prion protein from mice and to add prion proteins from different animal species. This is great because it allows us to model interspecies transmission in these animal species. And by doing this, we can analyze different pathological features of the disease. For example, deposition in the brain of the prion protein, tissue damage, etc. Unfortunately, these experiments are very long. Sometimes they could last for years. meaning that we will not have a rapid turnover of data, and the experiments will be expensive. So that will limit the amount of research that we could do. So something that also could be used are cell cultures. In here, we can also express prion protein from different animal species. And in general, cell cultures are easy to maintain, they are fast to propagate, they are easy to standardize, but for the specifically for prion research, sometimes they are difficult to infect. And unfortunately, they don't propagate all prion strains, and many of these prion strains are relevant. And we cannot modulate these processes in cell culture. So from a couple of decades, we count with other techniques that are cell-free techniques that allow us to investigate the conformational changes involving the prion protein in an accelerated and very efficient manner. So one of these techniques is the protein misfolding cyclic amplification, or PMCA. And in here, basically, what we do is to combine in a test tube a large quantity of normally folded protein with little amounts of the infectious particle. So when these proteins get in contact and we let them to incubate, basically happen, as I explained before, a recruitment of normally folded proteins into the growing aggregate. So these aggregates grow by the ends, and at some point, the conversion will become inefficient. So what we do, we break these aggregates and we generate new reactive ends. And we do this by sonicating these fibers. When these fibers are sonicated, we generate new free ends that will allow the additional recruitment of normally folded proteins into the growing aggregates. And by repeating this process of incubation and sonication, we can reach an exponential, we can obtain an exponential generation of infectious particles. So as you can imagine, this is great for diagnosis, but also is good to analyze several mechanisms involving prion diseases, as for example, interspecies transmission. And we can do it in an accelerated way. So in this figure generated by the group of Dr. Claudio Soto and Dr. Sandra Pritzko, what it was done is try to establish how useful PMCA is for study zoonotic potentials. Like again, in here, what they did, it was to take sheep scrapie and BSC from cow, and when you test the replication efficiency of these prions into an homologous system, so for example, using sheep scrapie in sheep PSC substrate, what it's seen is a good amplification of the infectious agent, a good detection of the infectious agent. Here, each signal is represents a positive detection. And the numbers in here represent the dilutions of the brain. So basically, we can detect brain material that is diluted 100 million times. So the amplification is great. For cow prions in an homologous substrate, using cow substrate, we can see that the amplification is even better. Now, if we try to analyze the zoonotic potentials in vitro of these prions, we can see that actually is reproduced what it has been widely accepted over the years. So for example, in a first PMCA round, we can see that sheep prions cannot induce the misfolding of the human prion protein. However, if we test cow prions into the human substrate, we can see that there is an induction of protein misfolding, although this induction is not as great as when cow prions are submitted to the homologous substrate. So this makes sense. And this result tells us that PMCA reproduced expected interspecies transmission of animal diseases in this humanized model. So we have experience working with interspecies modulation of prion diseases using PMCA. This paper that we published long time ago using hamster and mice is a proof of this, and relevant for the investigation of zoonotic potentials. The group of Claudio Soto in 2011 published this paper where he tested chronic wasting disease and the potential that it has to induce the misfolding of human prion proteins. And in here, he observed that it was possible, although CWD presented a strong barrier for inducing the conversion of human prion proteins. And in addition, it was also observed that maybe only some strains of the agent are able to do so. So we follow and expanded that research and actually using a grant that was funded by the Creutzfeldt-Jakob Disease Foundation in 2018, we generated 4 different chronic wasting disease strains, and we observed that these 4 strains had different potentials to induce the misfolding of the human prion protein. Okay, so we wanted to take this research further, and we wanted to analyze what happened with pigs. You know, pigs are an important component of human diet. So in that sense, we know also that the pigs can be infected experimentally by different type of prions, prions from cow, prions from sheep, and prions from deer. Now, it is important to highlight that no natural cases of pig prion disease have ever been reported. However, we also need to acknowledge that there are no surveillance programs trying to see the presence of prion disease in pigs. And of course, at present, there are no cases reported of prion transmission from pigs to humans. Anyways, we wanted to see, considering that pigs are susceptible of prion transmission from these different animal species, we wanted to see what are the properties of the prions generated in these animals, and also try to analyze the zoonotic potentials that they could have, and whether in the future they could represent a threat for human health. And what we observe in here is that the signature of the prion strains that is generated in the pig is different compared to the one of the parental strain. For example, in here you can see the bovine prion signature. It looks like this. This band that we call diglycosylated is the most prominent, and in the pigs, the monoglycosylated is the most prominent. Something similar happened when we have sheep scrapie infecting pigs. We see also that the signature profile changes after passage in this new animal species, and something also similar happened when we use deer prions to infect pigs. In addition, the study of the interaction between the different animal prion diseases and pigs is relevant in the context of chronic wasting disease, because as you may know, chronic wasting disease is the only disease that occurs in the wild. It's more uncontrolled. You know, we don't have it's more difficult to contain it. And for example, in here in nature, animals will be dying from chronic wasting disease. You know, they will be in the forest, you know, and some animals, like for example, wild boars, will come to their carcasses and they will feed from them. And when that happens, these animals already get exposed to infectious prions, and eventually they may get infected. And if that happens, and if these animals, for example, hunted and consumed by humans, it could be an extra problem to consider. And also, during the last year, it has been observed that the population of wild boars in the United States is increasing substantially, and in many areas, we can see a high density of wild boars and also high incidence of chronic wasting disease. So all this information leads us to the hypothesis of this project, and this is that BSE, scrapie, and CWD-exposed domestic pigs accumulate different quantities and strains of prions in peripheral tissues. We are specifically focusing muscles, each one of them with particular zoonotic potentials. We plan to interrogate this hypothesis in 2 specific aims. The first one is to identify and characterize PRPSC in brains and muscles of prion-exposed domestic pigs. And the second aim is to analyze the zoonotic potential of porcine adapter BSE, scrapie, and CWD prions. So the experimental design for this project is actually very simple. What we plan to do is to take cow prions, scrapie prion, and CWD prions, and when these prions were used to infect pigs, we plan to take the brain, the muscles of these animals, and to test them in pig PMCA and also in human PMCA. In the first one, we plan to assess both tissues and see what is the potential of the prions generated after infection the prions in pigs after being infected, exposed to BSE, scrapie, or CWD, and see how is the potential of these newly generated prions to propagate within the same animal species. And for the human PMCA, the idea is to test the zoonotic potential, specifically focused in mussels, that is the material that will enter the human diet. And well, as I mentioned, specifically what we want to measure are the propagation rates of the prions within the same animal species, to analyze the strain properties and see how diverse the strains that are generated in these animal species are. And again, we will focus in the zoonotic potentials, specifically in brain and in muscles. So one of the main difficulties at the beginning was to get a good PMCA system to amplify prions in pigs. And we were able to do it. This was the first part of this project. Basically, in here, what you can see is that we can get a good amplification for pig prions using pig substrate. And the technique is very specific because our negative controls didn't show any signal. So in order to test how the system is working in relevant samples, what we did, it was to take several samples from pigs infected with scrapie and sacrificed at different times. Specifically, we analyzed brain, retropharyngeal lymph nodes, and muscles of infected pigs. And as you can see in a first PMCA round, we didn't get any signal regardless of the good amplification that we had. In a second PMCA round, we were able to maintain our efficiency and specificity, and we were able to detect prions in some of the tissues that we were analyzing. So the technique is working well. It looks promising. We are ready now to jump to the next step of this research. Unfortunately, our lab was affected by the pandemic. We had to stop some of our work, and we were not able to receive some of the materials that we needed to do this research. However, we already received some of it. We are on the way to receive the additional ones. And our future directions are to establish the replication efficiency of BSE, scrapie, and CWD-derived pig prions in homologous substrate, again, focusing on brain and 3 muscle types, to compare the biochemical properties of PMCA-derived prions with original PRPSC, specifically focusing on electrophoretic mobilities, glycosylation profiles, and resistance to proteases, and finally to evaluate by PMCA the zoonotic potential of brain and muscles of BSE, scrapie, and CWD-derived pig prions. And we will do it in 2 ways. One, by normalizing by PRPSC quantities, but also by normalizing by mass of tissue. So to finalize, I would like to thank the people in the lab doing this work, to our collaborators for this project, but especially to the Creutzfeldt Jakob Disease Foundation for the generous support, and specifically to the CJD Foundation grant, to the International CJD Awareness Day Research Grant, to the Jeffrey A. Smith Memorial Grant, and to the Strides for CJD grant. Thank you.